Hello everyone. In today's video, we are going to talk about general principles of food preservation, asepsis, removal, and aerobic conditions, etc. At last, we are going to practice few MCQ questions, and this particular topic is important for all those who are preparing for FSSAI, CFSO examination, technical officer examination, or any other state food safety officer examination. Without wasting time, let's start the video. First of all, we need to know why there is a requirement of food preservation. As we all know that food is very important for humans because they provide us with the essential nutrients which are required for for carrying out the metabolism of human body. In the same way, microbes also require nutrients for their growth and for their metabolism. So if these food components they are available to microbes, they feed upon them, flourish, grow, and then decompose the food, and after that the food is unsuitable or unsafe for human consumption. Most of the foods are readily decomposed by microorganisms unless special techniques or methods are used for their preservation. So now let's see what kind of methods could be utilized for the preservation of foods. The first method is asepsis. Asepsis is also known as keeping out microorganisms. If there is a protective covering about the food, microbial decomposition is delayed or prevented. Example of such coverings are the shells of nuts, the skin of fruits and vegetables, and cells of eggs, etc. It is only when the protective covering has been damaged or decomposition has spread from the outer surface that the inner tissues are subjected to decomposition by the microbes. Packaging of food is also widely used application of asepsis. The covering may range from a loose carton or wrapping, which prevents primarily contamination during handling and thus protects the processed contents from contamination by the microbes. The next method is removal of microorganisms. Removal of microorganisms is not very effective in food preservation, but under special conditions, it may be helpful. Like removal, it may be accomplished by means of filtration or centrifugation or washing or trimming, etc. Filtration is only successful method for complete removal of microorganism and its use is limited to clear liquids. While centrifugation is applied to, it is not very effective, uh, but not all the microorganisms are removed in centrifugation, while in sedimentation, it is usually used for the treatment of drinking water, but is insufficient by itself. When centrifugation is applied to milk, the main purpose is not to remove bacteria, but to take out suspended materials. Although centrifugation at high speed, it removes most of the spores. So filtration is only successful method for complete removal, and centrifugation or sedimentation, they do not provide very effective result. The next method is maintenance of anaerobic condition. So, uh, completely fill evacuation of a complete fill evacuation of the unfilled space, or we can say that the replacement of air by carbon dioxide or by an inert gas such as nitrogen will bring about anaerobic condition, and because of the absence of oxygen, the microorganism would be unable to germinate or grow, and this will help in preserving the food substances. Next methods are use of high temperature and use of low temperature. These two methods are widely used, and we all know the methods of freezing and refrigerations. They are used for inactivation of microorganisms or slowing down their growth. Next method is drying. Drying is also a method which is being traditionally used from ancient period of time, and it includes the tying up of water by solutes, and because of unavailability of water, the microbes they could not grow. Next is use of chemical preservatives, irradiation, mechanical distribution of microbes, such as by the grinding, by high pressure, etc. Although it is not industrially used. Next is combination of two or more of the above methods. So combining one or more of the above methods would really provide or give a very effective result. Next, coming to the principles of food preservation. So basically, there are three principles: preservation of or delay of microbial decomposition, prevention or delay of self-decomposition of food, prevention of damage because of insects, animals, mechanical causes, etc. Coming to the first one, prevention or delay of microbial decomposition. It is done by keeping out microorganisms, that is asepsis, which we have already discussed, by removal of microorganisms such as filtration, by hindering the growth or activity of microbes, by using low temperature drying, etc., and by killing the microorganism by the use of heat or radiation. Next is prevention or delay of self-decomposition of food. It is done by blanching. Blanching is using blanching, we can inactivate the food enzymes which is responsible for their degradation. And next is preventing or delaying purely chemical reactions. For example, prevention of oxidation by the use of antioxidant. And last one is prevention or damage because of insects, animals, etc. And all these methods are used to control the activity of microbes. Next is the growth curve of microbial culture. So this is a growth curve from this curve is divided into phases as it is indicated in the figure. The first one is the initial lag phase, which is from A to B, during which there is no growth or even a decline in number. The phase of positive acceleration, that is from B to C, during which the rate of growth is continuously increasing. And the logarithmic or exponential phase of growth from C to D, during which the rate of multiplication is most rapid and constant. The phase of negative acceleration, that is from D to E, during which the rate of multiplication is decreasing, and the maximal stationary phase, that is from E to F, where number remains constant, and the accelerated death phase from F to G, and the death phase or phase of decline from G to H, during which number decreases at a faster rate, and the last one from H to I is the survival phase, during which no cell division occur. So mainly you need to remember the initial lag phase, then the logarithmic phase during which multiplication increases, then the stationary phase during which the number remains constant, and the death phase during which the number starts to decline. So this is a table which shows the classification of preservation factors. What is the mode of action, what are the preservation factors used, and how it is, which method or mode of achievement is used. The first one is inactivation of microbes by using heat or radiation, and the process involves are pasteurization, sterilization, etc. Next is inhibition or slowing of growth of microbes, which is done either by chilling, freezing, drying, addition of salt, sugar, fermentation, etc. Next is restriction of access of microbes to products by using packaging, aseptic or clean handling, and by using microstructure control and decontamination, etc. You all can go through this table. It is very useful. So now we are going to solve some multiple choice questions. The first question is, asepsis is reducing the population of microorganisms, identifying methods of destruction, sterility check, or keeping out microorganisms. So the correct answer would be, I have already said in the video that asepsis is keeping out microorganisms. So the correct option would be option D. Next question is, delay in microbial decomposition can be achieved by asepsis, filtration, radiation, or all of the above. So all these above mentioned processes can help in delay in microbial decomposition. So the correct answer would be option D, all of the above. Next is prevention of self-decomposition can be accomplished by option A, blanching, option B, washing, option C, chemicals, option D, heat. So the correct would be option A, blanching, because blanching helps in inactivation of microbes and help in prevention of self-decomposition. Next is, which of the following is not a preservation factor? The first one is to restrict oxygen, next is radiation, next is heat, then option D is mechanical damage. So the correct option is mechanical damage. The next question is the rapid and constant rate of multiplication of an organism occurs during the lag phase, exponential phase, stationary phase, or the survival phase. So the correct answer would be exponential phase, option B. Microbial decomposition of foods can be prevented by decontamination, killing microbes, scalding, or none of them. So the correct option would be killing microbes, option B. Restriction of access of microorganism to a product cannot be achieved by using heat, HCl, H2O2 or H2O. As answer would be H2O. The widely used application of asepsis is clean handling, decontamination, packaging, or radiation. So the correct answer would be option C, that is packaging. The next question is heat is used to inactivate microbes, inhibit growth of microbes, kill microbes, or restrict the growth of microbes. So the correct answer would be inactivate microbes, option A. Bacteria proof filter is made up of sintered glass, di- at diatomaceous earth, unglazed porcelain, or all of the above. So the correct answer would be all of the above. That was all about the topic. Thanks for watching. And if you like the video, please do like, share, and subscribe to Food Tech Shiksha.